Hello everyone and welcome to this webinar co-produced by the CRISPR Journal and Genetic Engineering and Biotechnology News. Our special guest speaker today is Professor Helen McCarthy. She'll be taking us on a stealth mission, a novel mRNA vaccine delivery system. Our thanks to TriLink Biotechnologies for sponsoring today's webinar. I'm your host for today's event, Kevin Davis, the founding executive editor of the CRISPR Journal, now in its 5th year of publishing, as well as our new peer-reviewed journal, Gen Biotechnology, which debuted in 2022. I'm also the author of the book Editing Humanity, The CRISPR Revolution and the New Era of Genome Editing, out now in paperback. Our sponsor today is TriLink Biotechnologies, part of the Maravai Life Sciences Group. TriLink is a CDMO for overcoming challenges in the synthesis and scale-up of nucleic acids, NTPs and mRNA, including its proprietary CleanCap mRNA capping technology. TriLink offers cGMP and general mRNA, oligonucleotide and plasmid manufacturing to support therapeutic, vaccine and diagnostic customers. It's a great pleasure to welcome our guest speaker today, Professor Helen McCarthy. Helen is a professor and associate pro-vice chancellor in the School of Pharmacy at Queen's University, Belfast in Northern Ireland, where her group specializes in the development of non-viral delivery systems for nanomedicine applications. She's also the founding CEO of Phion Therapeutics, a Belfast-based biotech developing a pipeline of therapeutic and prophylactic vaccines focused on viral infections and oncology. This is the final episode of a 4-part webinar series generously supported by TriLink on the latest advances in mRNA research and technology. We hope you've had a chance to watch the other 3 episodes in this series. If not, you can view them on demand. Those presentations were by Norbert Pardee, Edward Miraco, and Philip Santangelo, and you can see them on demand and find them on the Gen multimedia page on the website. Before I hand over to Helen, please enter your questions for the live Q&A session at the end of her presentation. Please just hit the Ask a Question button at the top of the screen and submit. We'd love to have a lively discussion later on today. All right, let's get today's presentation underway. Helen, it's great to see you. Thank you so much for joining us, and we're really looking forward to your presentation.
Thank you very much for that introduction, and thank you to everyone who has tuned in to listen to this presentation today. It's an absolute honor to be able to talk to you about our drug delivery technology and how we're using that for stealth-like delivery of messenger RNA vaccines. Today in this talk, I'm going to explain to you who Phion Therapeutics are. I'll also talk a little bit about our technology and how it works, the key advantages of the technology, and then we'll look at some examples both in infectious disease and in oncology. So Phion Therapeutics is a spin-out company from Queen's University, Belfast, from the School of Pharmacy there, and we spun it out in 2017. It is based on the research of myself, where I hold the chair of nanomedicine within the school. There has been a lot of underpinning research that has gone behind this technology, many years, in fact, and a lot of experiments have underpinned what you see today. We've published a number of articles and we've secured a lot of funding from key partners, both in the UK and also from Ireland and the US as well. We work with a lot of partners currently right now. Invox Pharma is our lead investor. We've also had a lot of funding from Innovate UK, a lot of support from Invest Northern Ireland for young businesses, and we work very closely with the Catapults, which I'm sure many of you will have heard of. And these are really good not-for-profit organisations up and down the UK that really want to help academics and young businesses commercialise their technology. So this is our team. You can see myself there. My Chief Operating Officer is Darren Neeson. He is a lawyer by trade and actually left Queen's to come and join the company, which is great to have a lawyer in-house. John Fox is our chairperson. He has many years experience which is what you need when you're trying to start a business. And then Ben is the director of Invox Pharma. George Orphanides is ex-AstraZeneca. And frequently what we have found in our young business is that we need to dial into a number of different experts. As an academic, I hold my hands up. I cannot do absolutely everything that is required. So we tap into a number of regulatory, clinical, and safety experts, as you can see on the slide. And that really is such a learning curve for me personally, and also for our team here in Belfast. To have that expertise and knowledge from people who've done it before really does make all the difference to us. So over the course of the last 5 years, we have developed a pipeline of messenger RNA vaccines. We also have one small molecule vaccine in there, which some of you may identify. We have a number of products. PTXV1 is our mucosal HPV vaccine. And the vaccines that are for oncology indications are given intravenously. The vaccines that are for infectious disease are given intradermally. A lot of these vaccines have been supported by Innovate UK. You can see there we also have our COVID-19 vaccine in addition to a Crimean Congo hemorrhagic fever. The Innovate UK, on behalf of the UK government, identified a number of diseases which align very closely with the World Health Organization that they were concerned about that could become diseases of concern. And Crimean Congo hemorrhagic fever is one of them, and we are working on that at the moment. So what I plan to do today is really talk to you about two examples of the technology that we use both for PTXV1, our HPV-driven therapeutic vaccine, and PTXV4, our multi-antigenic COVID vaccine. Okay, so let's talk about what the technology is. What is the RALA platform? Well, essentially, RALA is a 30 amino acid peptide, and you can see the sequence of it on your screen right there. And the idea is that we're using that peptide to deliver messenger RNA. We all know that messenger RNA will not be able to enter cells without a great delivery system to get it there. And that's certainly been the case with the COVID vaccines, with the lipid nanoparticles delivering that messenger RNA. As we know, the lipid nanoparticles have been really great on one hand by facilitating the messenger RNA vaccines, but they are also not without their limitations. I have been working for many years developing this peptide for a number of applications. Indeed, we published articles that we'd use this peptide to deliver both DNA and messenger RNA vaccines in 2017. You will notice on the peptide itself, there are a number of arginine residues nicely spaced out, and in between them is spaced alanine, and then we have our hydrophobic leucine. And essentially what this does, it condenses the nucleic acid cargo into really small nanoparticles. The arginine head groups, you have guanido groups on those, and they form the hydrogen bonds with the anionic cargo. The real key advantage of this technology, however, is that it is not recognized as foreign. And when you are building a vaccine or when you're trying to modulate the immune system in any way, you really don't want your delivery system itself to cause an immune response. So on the basis of that, you can then give these nanoparticles multiple times. And to date, we haven't been able to find that the RALA peptide is not recognized as foreign. This really isn't a huge surprise because the RALA peptide is made of natural L-amino acids. And if we think about it, proteins routinely move in and out of their cells all the time. But because of that, the immune response that we get with this technology is quite interesting. We have a complete bias towards the CD8 Th1 response. And that is something that we've identified time and time again. And we can use that to an advantage, particularly when it comes to our cancer vaccines. We haven't been able to find any RALA neutralizing antibodies, even after multiple injections. And we've published on this again in 2017. We're not restricted by the size of the cargo because it's based on the charge density of the cargo, and we can deliver as many different types of cargo as we want. So for example, we can deliver a strand of siRNA with messenger RNA or DNA with microRNA, and we can deliver multiple messenger RNA that code for different things or microRNA. It's also stable, so we lyophilize the product and we get stability at room temperature for greater than 18 months. We haven't measured it any longer than 18 months, but I would imagine that it would be stable much longer than that. And what we do is we cryoprotect the nanoparticles so that upon reconstitution, they are isotonic and can be delivered safely then. And we also use an automated scalable process, and we have scaled up to commercial doses. This is something that I think many young scientists who are thinking about delivery systems and technologies like this should bear in mind. How easy is it to make and manufacture some of these new, more sort of advantageous biological platforms? Is how easily and how quickly can you manufacture it? So how does it all work? How do we put it together? Well, essentially you take your anionic messenger RNA, can be any nucleic acid, and we add our cationic RALA peptide to it. We identify what the ratio of peptide is to the messenger RNA, and we use the Precision Nanosystem suite of machines. Here in the slide, you'll see there's the Ignite machine, and that gives us an automated and controllable mixing system. So we can control the flow rate, and we can ensure that we get reproducibility in our nanoparticles. As I said before, those of you may have noticed that there is a cysteine residue at the end of the peptide. When the peptide intercalates with the cargo like this, the cysteine residues also help to stabilize the nanoparticle by forming disulfide bonds. We end up with a nanoparticle that is the right size and charge to enter the cell via endocytosis. There really isn't anything new in that, except to say where TAT, peptides, etc., would have got trapped in the endosome, our peptide is exceptionally good at getting out of the endosome. So we enter the cells typically via clathrin-mediated endocytosis, and we proved this in a publication in 2014. And once it gets inside the cell, it is trapped within the endosome, and what you find happens then is there's a drop in the pH. The drop in the pH then makes the peptide more alpha helical. It fuses with the endosomal membrane and it releases the cargo. And the cargo then can escape through an endosomal pore, and the cargo gets into the cytoplasm where it becomes more bioavailable. The more bioavailable your cargo, the greater the therapeutic effect. Also, what is quite important is in studies that we did with cell and gene therapy catapult, we showed very clearly that whenever we deliver the nanoparticles to the cell, we don't change the receptor status of the cell at all. So when we use nanoparticles that have a reporter cargo or an inert cargo, we are not changing the cell itself. And that's really important, because if you do change the cell, you want that to be a result of your cargo, not your delivery system. Okay, the other thing that is really interesting that we found out with this technology is that we can encapsulate nearly 100% of our cargo. Our assay isn't sensitive enough to go to 100%, but we do encapsulate 99.71% of our cargo. And we did this through ionic exchange chromatography, where you can see whenever we put water and our nanoparticles through a column and we put messenger RNA through the column, you can see very clearly that the messenger RNA is detected and passes through the column. You can see that the RALA messenger RNA nanoparticles do not pass through the column. There's no free messenger RNA to pass through the column. And in fact, they're identical to water. And whenever we quantify this, we get a really high percentage of messenger RNA included in our nanoparticle. This again is very important because we don't want to lose any of the product, because messenger RNA is very expensive and it does cost quite a lot of money to make your final products with this. On the back of this though, because we don't use any solvents in our nanoparticle process, we don't need tangential flow filtration once we make the nanoparticles. And again, this does save a lot of time and money and means that we only essentially need sterile filtration. So I talked about the microfluidic machines that we use. You can see we used to start manually, as all scientists do. And then once we formed the company, we moved into microfluidic systems. And the Ignite will take you up to 20 mils, and the Blaze machine can take you up to 10 liters. We use the Blaze machine to formulate these nanoparticles for toxicology. And then there's a GMP continuous production machine that we put in a CDMO to make the product for clinical trials. You can see that in terms of our manual formulation, we're getting really nice Z average particles, but they improve upon use of the Ignite and improve further on use with the Blaze machine. And we have PDIs that range from 0.2, which is a pretty homogeneous population, down to 0.121. So again, you can see the distribution gets tighter and narrower the more we scale up this product. And that's exactly what we want to see, because we have to be able to manufacture this if we want this to become a vaccine that we use in the clinic. This is an example of following lyophilization. You can see the vials there on the left-hand side. The RALA nanoparticles can be stored at room temperature. And messenger RNA functionality is retained for at least 18 months. We have a nice transmission electron micrograph, and this is actually of nanoparticles that contain 3 messenger RNA within our PTXV4 product. And the fact that we're not seeing any agglomeration, which supports the low polydispersity index, is really encouraging for us. And for somebody that's been trying to get discrete nanoparticles for many years, it's very pleasing to see that we have this formulation that gives results such as this. One of the questions that we always get asked is, okay, so you've made the nanoparticles, they're very stable and lyophilized. How do they function whenever you add your sterile water to them, i.e. what's the in-use stability? Because a healthcare professional may add the sterile water to it, but how long will it remain functional over time? So we tested this using lyophilized RALA GFP messenger RNA nanoparticles, and they were reconstituted in ultra-pure water. And then we tested the functionality in our NCTC cells. And what we found was that these nanoparticles retain full functionality for up to at least 96 hours. We haven't taken it beyond 96 hours. We figure that a healthcare professional will probably administer the vaccine within that time period. So we will test this further to find the limit of stability, but something that I definitely learned was the importance of in-use stability when you're making a new product. So what does it look like when we put the whole process together? Well, we can scale up the clinical doses again, and once you make your messenger RNA and get your nanoparticles, we add our cryoprotectant sugar. We actually use a 0.2 micron sterile filtration, and it's used. We use a vacuum pump for this. We then lyophilize, and there we have our product. So all in all, it takes less time, and there's less wastage, and it is more cost efficient. We have a number of ongoing studies where we're looking at further stability of these RALA messenger RNA products, and we're looking at how they fare under various ICH zone 1 to 4 conditions. Because again, some of our vaccines that we're making will go to very hot regions of the world, and we want to make sure that they retain stability and functionality. So I've told you a little bit about the technology, how we make the nanoparticles and how we can scale them up and the importance of that. What I'd like to focus now on is the application of it for 2 of our vaccines. Our first vaccine is known as PTXV1. This was supported with funding from Innovate UK Biomedical Catalyst Award in the first instance, and then Vion have subsequently taken this on to develop this towards the clinic. This is for mucosal HPV and HPV-induced cancers. So really we're focusing on HPV-16-driven cancers. And one of the questions we get asked is, how do our nanoparticles work as a vaccine in vivo? And why are they different? And why does the RALA platform make them different? This was a landmark study that we did in partnership with Ghent University Department of Immunology in 2017. And what you can see here is that these mice on the left-hand side are genetically engineered, and they're genetically engineered to produce luciferase under the control of the interferon beta promoter. So when we get production of interferon beta, we get production of luciferase. Now, interferon beta production, when you're dealing with nanoparticles, is not your friend. It is not your friend because it is an innate response, and it will clear your nanoparticles out pretty quickly, so you're not getting the maximal therapeutic effect. What you can see, I hope, is that when we use uncomplexed messenger RNA, there's a high signal of luciferase. When we use a DOTAP liposome, we still get production of luciferase. But there, when we have the RALA messenger RNA nanoparticles and we inject these intradermally, you can see that there is no presence of luciferase. This indicates that there's no production of interferon beta. If you want to create a really strong CD8 Th1 vaccine, focusing on getting production of interferon gamma is what you want rather than interferon beta. So that's all very well and good, and you may ask the question, okay, maybe your nanoparticles were undetected, but maybe they didn't go in and do the job. So we went on to do studies where we labeled the messenger RNA with Cy5. Cy5 is a fluorescent label, and we delivered these to the mice using RALA and also with naked Cy5 messenger RNA on its own. We gave the mice intradermal injections, and we opened the mice up 2 hours later to see where the Cy5 signal was being found. You can see very clearly that in the Cy5 only messenger RNA group, there is no signal in the lymph nodes, whereas in the RALA Cy5 messenger RNA nanoparticle, you can see a clear signal in the lymph node at 2 hours. Now, the cynics amongst this may ask the question, okay, you've got a Cy5 signal there, but is that actually an immune response, or is that the draining lymph nodes that the messenger RNA has just migrated to? Well, the answer to this is we took out the lymph nodes and we had a look to see what cells they were getting into. And what we found was, as you can see in the blue in the first panel, that the RALA Cy5 messenger RNA nanoparticles were getting into the antigen-presenting cells, the CD11C plus cells, in a very healthy percentage of them, averaging there about 8%. And that remains so over 48 hours, and then it drops down at 96 hours. So it's getting into the antigen-presenting cells in the lymph nodes, but the question is, has it been actively transported there through migratory antigen-presenting cells, or is it just drained there and got into the lymph node resident cells? So we pulled these cells apart further and had a look to see, and I'm pleased to say that it was actively being transported there by migratory cells, and it wasn't the resident cells that were taking up the cargo. So we can be confident that whenever we give our nanoparticles through intradermal injection, that the migratory cells, migratory antigen-presenting cells, are taking this up and transporting this then to the lymph nodes, whereupon basically these antigen-presenting cells can talk to the T cells. And so we then wanted to apply this technology once we knew how it worked using the E6, E7 messenger RNA for human papillomavirus 16. Now, as we know, HPV-driven cancers are on the rise, and even though there is a prophylactic vaccine that is given out to young females and males in the UK, it's actually the disease itself, and HPV-driven head and neck cancer is the most sexually transmitted disease right now within the UK. And there is a higher incidence of this disease in areas that have low socioeconomic status. And so there really is an unmet need to have a therapeutic vaccine for this. And so we know that with respect to cervical cancer, that HPV is responsible for around 90% of these diseases, but we're also finding that in the clinic, it's responsible for around 70% of the head and neck cancers that are coming into the clinics, particularly in the northern half of the UK. The E6 and E7, essentially, E6 interacts with P53, and as a result of that, you get a loss of the P53 functionality, while the E7 binds to the retinoblastoma tumor suppressor, and essentially it potentiates cell cycle proliferation. So both of these genes, antigens, have an impact on the natural sort of regulatory function of the cells within cervical cancer and head and neck cancer. We also chose these genes because we wanted to differentiate our technology from those that have tried before with these genes with either lipids or electroporation. So the first thing that we had to do was we knew from earlier studies that we were getting a bias towards a CD8 Th1 response, but we had to prove it with these antigens. And this is where TriLink have really helped us, both in making sure the design of our antigens was really where we wanted it to be, but also in the production of really high quality messenger RNA. So for these studies, we gave the mice a dose at day 0, and another dose 21 days later. And we gave a 20 microgram dose of the RALA messenger RNA each time. And at 42 days, we took out the spleens and we cultured the splenocytes, and then we looked to see if we were getting an interferon gamma response. And as you can see in the top panel, the fluorospot indicated that we were getting an interferon gamma response, in fact, averaging more than 200 spot-forming units per 250,000 splenocytes. In terms of then which cells were producing this interferon gamma response, and also markers of interleukin-2 and TNF-alpha, we looked at both CD8 cells and the CD4 cells. And we were actually really shocked whenever we found the results of these, because you can see very clearly that the CD4 cells in the bottom panel are not producing any of this interferon gamma or interleukin-2, or indeed TNF-alpha. If you compare to the top panels, you'll see that all of these cytokines were being produced by the CD8 cells. So we were getting a very antigen-specific CD8 T-cell response. We then wanted to have a look to see, okay, so what are the extracellular cytokines produced by the splenocytes? And we can see that whenever we put the splenocytes, cultured them with the E7 overlapping peptide, that we got, again, a very strong interferon gamma response, and we didn't get any production of interleukin-4, which is an indicator of a Th2 response. So again, everything is indicating that we're heading towards Th1 CD8 response. I guess the question we then asked ourselves was, how does this translate to both a prophylactic effect, but also a therapeutic effect? So what we did here was we gave 2 doses of the vaccine, again, 21 days apart. We decided to stick with the 3-week between each dose. And then we gave these intradermally into the ears of mice, because that's where you get a higher percentage of antigen-presenting cells. We then challenged the mice 7 days later with TC1 tumor cells, which are the typical model that are used for HPV-driven cancers. And what we also did was 80 days later, we re-challenged the mice with TC1s. And that was because we wanted to see if we could get T-cell memory. So what we found was that we had 8 mice in each group. The black lines you can see are the naive mice. The blue line are the mice that were vaccinated. And you can see that none of the mice developed either tumors in the first instance, nor did they develop tumors upon re-challenge. And we used age-matched naive mice again whenever we re-challenged to see how the controls would grow. And what this tells us is that our vaccine is pretty effective in a preventative regimen, but also that it has long-term T-cell memory. And that's exactly what you would want to see in a messenger RNA vaccine. Indeed, we also took out tumors where we could and identified and looked to see for markers. Obviously, there was no tumors in the mice that didn't get any tumors, so we took out the spleens and were able to identify that there was a CD62L upregulation and that the T-cell memory was evident. The question then is obviously, okay, prophylactic is pretty easy to prove. So what about therapeutic vaccination? So what we did here in the therapeutic vaccination is we gave the mice tumors to begin with, and then we weren't exactly sure what the optimal dosing regime was. So we gave doses 8 days later, 7 days later, and 28 days later, and then we re-challenged. And what we found from this study was that we only got around a 50% response rate. Now, we gave, again, naive mice are in black, age-matched naive mice are in the light grey. Our vaccine is PTX-V1, and we also used a control messenger RNA called PTX2. And you can see that some of the mice, about half the mice, developed tumours in the vaccinated group, and 50% of them didn't produce tumours. And what that told us was, yes, our vaccine works quite well, but it doesn't work well enough. So these were, again, mice that were vaccinated intradermally. And so on the back of that, we said, you know, we need to get into a higher number of antigen-presenting cells really quickly. And we need to do this because with an established tumour, we want to mount an immune response as quickly as we can. With prophylactic vaccination, you've got time to develop immunity before you get infected with whatever disease it is. But with a therapeutic response, you need to get into a high percentage of antigen-presenting cells as quickly as you can to mount that T-cell CD8 response. So in this study, again, we used TC1 tumors. We gave the mice tumors at day 0. 4 days, and then 3 days, and then 4 days apart, we gave dose 1, dose 2, dose 3. Again, this was 20 micrograms of our vaccine delivered intravenously. And the results that we found were pretty astonishing. Essentially, only one of our mice got tumor, as you can see, and 88% of our mice didn't get any tumors at all. And they are currently being rechallenged, and none of them that have no tumors before the rechallenge, they have not got tumors after the rechallenge. So we asked ourselves the question, okay, what is going on? And so to answer that, we gave the vaccine again to mice, and we pulled out essentially the spleens after 1 hour and after 24 hours. And what we found is we're getting into a really high number of CDC2 cells, macrophages, resident CD1 cells, and migratory CD1 cells. So on all fronts, we're getting a really high, greater therapeutic response compared to intradermal delivery. And it is critical that we have this cellular response as quickly as we can. So where we're at now with this study is we've done another tweak to the formulation, and studies are ongoing where we've flatlined, so none of our mice are getting any tumors, both in the TC1 model and the C343 model, which has all the antigens for HPV-16. And whichever one of these formulations is the best, that's the one that we're going to take into the clinic next year. So in order to take it to the clinic, one of the first things that we're working on right now is we're working on the toxicology. And so we've already done the species selection with toxicology. We've had advice from the MHRA, and we're doing our toxicology in rats. So as soon as we pick this final formulation towards the end of this year, the toxicology will start in the new year. And we'll then look at injection sites, the pathology, we'll look at the organs, we'll look at the biodistribution, and we'll look to see if we've got any histopathology. So exciting times for the company. It's our first time into tox, and we're really looking forward to seeing the results. One of the things that I have learned throughout this process is that it's important to take your time and make sure you get your preclinical candidate just right, because once you go into clinical trials, there really is no going back to reformulate, etc. So we are making sure that we get the best formulation we can before we commence the tox in the new year. With respect to the clinical trial, we're working very closely with Aptus Clinical, and we're developing a modular clinical trial plan. And by that I mean we're going to look first at the first module, and on the results of that, we'll take the decision as to whether we'll go into the second, the third, and the fourth module. And the idea is that we're going to look in module 1 at the safety and the pharmacodynamics in advanced HPV tumors in patients. So we're going to have a look at 2 different vaccination schedules. We'll have a look at a relatively small cohort of patients, and we just want to make sure that the vaccine's safe before we move on into look at dose finding for patients with HPV-driven cervical intraepithelial neoplasia. So module 2 is where we look at dose ranging in patients that have SIN 2/3, and module B is where we look at the proof of concept then in patients once we know the dose that we're going to use in part A. Based on the results of that, then we'll look at a proof of concept where we will look at patients with HPV-driven invasive cervical cancer. So from module 2 to module 3, we're getting more advanced in the cancer. And then from module 3 to module 4, we're looking at patients that have HPV-driven head and neck cancer. And again, each of the results from these modules will inform us as to what we're going to do in the next stage of the clinical trial. Obviously, we don't do this in isolation. We get a lot of support, both preclinically and also in the CMC department and also in the regulatory and clinical department. And I think, again, It's a really exciting network whenever you're developing products to the clinic, that you work with a number of partners that help you get there. So that's an example of our HPV V1 vaccine for therapeutic-driven diseases. What I'm going to talk about now is our triple messenger RNA vaccine for COVID-19. Again, we're using the same RALA messenger RNA vaccine platform, except this time we're doing it in a prophylactic model, and obviously I don't need to introduce COVID-19 to any of you. One of the reasons why we deliver these vaccines intradermally is because there are a higher number of dendritic cells that are present in that intradermal layer. And if we can get our vaccine into those antigen-presenting cells, then we can get a really good, strong immune response. And you'll remember from the earlier data, when we looked at a preventative regimen for HPV-driven cancers, that it worked very well. A lot of people have asked me about, well, are you concerned about how you're going to actually give the intradermal vaccine? And do you need specialized training? There are a number of devices on the market that we are evaluating at the moment, from PharmaJet to NanoPass, again to UNeedle, to see which one is the best for us, and also which one do we have least wastage in, and which one would our healthcare professionals be happy to use. I don't know if you're aware, but the monkeypox vaccine that is being rolled out across the UK has recently moved to a microdosing regimen, and that microdosing regimen is essentially intradermal delivery, again, because there's a recognition that there are more antigen-presenting cells there that will mean a smaller dose will go a lot further. So what is PTXV4? This is messenger RNA encoding multiple SARS-CoV-2 antigens in a blended ratio. So for these vaccines, we decided that we would look both at the spike, the membrane, and the envelope antigen. And the reason that we chose the membrane and the envelope antigen is because the number of reported mutations was significantly less than the spike mutated antigen. Indeed, it's very difficult for anybody to keep up with the number of mutations in the spike antigen. And so we decided that we were going to look at the membrane and the envelope in tandem with the spike. The theory being, if the spike does mutate, you still have the membrane and the envelope antigens that can help the activation of your vaccine. Exactly the same method of producing it. We use the messenger RNA, we use 3 in a blended ratio, and we add our peptide to that, use the microfluidics, form the nanoparticles, lyophilize them, and then you have your product. So what we looked at, first of all, was what is the immune response to the vaccine itself? Again, we give a dose at day 0. We give another dose of our messenger RNA vaccine at 21 days later, and we analyzed at day 42. A lot of other preclinical data shows that they have around 200 to 300 micrograms of mil for spike-specific IgG in mice, which you can see is the gray shade on the diagram. What we found was that both the RALA spike and the RALA SME nanoparticles, we call them SME because there's 3 antigens in them, they provoked an anti-spike IgG accumulation in the serum of mice, and indeed really did give quite an encouraging amount of IgG in comparison. You'll see there's a compound effect whenever you add in both the membrane and the envelope, so you end up with a really strong IgG response against 3 different antigens. This is a really good template because while COVID may be becoming more attenuated and less virulent than it was before, there's still an opportunity to put in multiple antigens and use this platform for many different infectious diseases. We also looked at the safety in BALB/c mice. Again, we vaccinated the mice and we had a look to see what the weight of the mice was over time. We also looked at the creatine, the AST, the kidney and liver enzymes, and the ALT activity. The gray bars are where sort of the normal would be for the enzymes at the bottom. The gray bars in the weight indicate that you wouldn't want any of your mice to lose more than 20% of your weight. And you can see over a time course that all of our mice essentially gained weight over that time period, and none of them seemed to drop anywhere near that 20% weight loss. We've done a number of other safety studies, but I don't have time to go through them all with you today in this presentation. One of the things that we did with BioClinics was we did a SARS-CoV-2 hamster challenge study. So we gave the mice the vaccine at day 0, day 21. We then challenged these golden Syrian hamsters with the original strain of SARS-CoV-2. And what you do then is you monitor these hamsters over a 4-day period, and you look at a number of key attributes, and then you terminate these hamsters for histopathology and a number of different analyses. What we found was that where these hamsters get the SARS-CoV-2 is in the nasal cavity. And so you can see the vehicle only, there was still presence of SARS-CoV-2 in this nasal turbinates, and also in the vaccine itself, in the vaccinated hamsters themselves. And this isn't unexpected because that's the site of administration of the virus itself. But what we did find very interesting was when you look at this 5-day study from day 42 to 46, that in the vehicle-only hamsters, you can see that gradually over time, there is an increase in the titers of the SARS-CoV-2 that basically get over a certain threshold at day 46 in the throats. What we can see in the vaccinated mice is that there really isn't a migration of the disease from the nasal passage down to the throat. So again, this is pretty good, and it tells us that our vaccine is working strongly. We then wanted to see, okay, the vaccine isn't getting from the nasal cavity to the throat, or sorry, the disease isn't getting from the nasal cavity to the throat, but is it getting down into the lung tissue? And the answer is the disease is getting into the lung tissue, but at a much less amount of the disease compared to our vehicle only. So you can see our vehicle only there has a log to the 10 of around 6, whereas our average is 2-fold log less that we find in the lung tissue. And indeed, in a few of our hamsters, it was virtually undetectable. So we're awaiting a number of pathology results from this study, but very excited to see that it actually works against SARS-CoV-2 in these golden Syrian hamsters. So just to summarize, when you look at our RALA messenger RNA vaccine platform, we've split it into 2 arms in the company. We have the oncology arms, which are delivered intravenously. Again, the immune system, the immune response that you want to get dictates the route of administration. And on the other side, we've split infectious disease to be delivered intradermally. Again, you've got time for the immune response to build up, so there's no need to hit such a high number of antigen-presenting cells in the first instance with infectious disease. The key advantages either way are that we don't get an innate immune response to our vaccines. We can use this technology for both prophylactic and therapeutic models. We have a multi-layered IP position. We have figured out the scalable formulation process. We have a very stable vaccine at room temperature, and we can get a strong CD8 response, which I think is really key whenever you're trying to clear particularly some oncology indications. And finally, all of our safety studies so far have come out really exciting and also haven't given us any cause for concern. I'd be very happy to take any questions. I'd just really like to thank you for taking the time to listen to our presentation and to find out about this exciting technology. Thank you.
Thank you very much, Helen McCarthy. A brilliant presentation, and thank you all for staying around as we enter shortly our Q&A discussion. A reminder, we'd love to have your questions to pose to Helen over the next 10 or 15 minutes, so please enter them into the Q&A box. Okay, please submit your questions for our Q&A session. We'll be archiving today's webinar, so you can view it on demand along with the other 3 installments in this TriLink series, and you can share that with your colleagues or view on replay. All right, I think it's time now for us to take some live questions from the audience, so just give us a couple of moments as we transition into the Q&A session.
It's a great number of questions from the audience. Why do you use different routes of administration for oncology versus infection indications?
Yeah, so that's all related to the immune response that you want. So if you think about it, and the pandemic is a really good example of this, if you vaccinate to prevent disease, you want to build up your immunity over time. So it's more of a gradual approach. So when we deliver the RALA intradermally, we still get into antigen presenting cells, but there's not the same urgency to get a really strong, fast CD8 response. With a therapeutic vaccine in cancer, you essentially want to make sure that you mount a response very quickly against the tumours. So in addition to getting into the antigen presenting cells that you would find, let's say, in the spleen, there are also circulating macrophages. There's antigen presenting cells that can be found in loads of locations. And so you get a much faster and a higher activation of your T-cell response. And that's the response that you really want to hit your cancer cells with.
Great. Is the RALA peptide the last word in mRNA delivery in your hands, or are you still iterating on that sequence?
So I think there's this misconception in the field that one delivery system will do everything, and I don't think that is the case at all. I think it very much depends upon the biology of the disease and what it is you're trying to do. We know that our RALA peptide delivers cargo into antigen-presenting cells and other cells to produce protein virtually undetected. And so the advantages of this are pretty specific for vaccines, because the immune response that you get is purely from the cargo itself. And is it the last word in delivery? I can't really answer that, but we have finished changing. We're not going to change any more sequences in this RALA peptide. We think that it is as good as it can be for the delivery of messenger RNA for vaccines. That's not to say that we couldn't at some point, and we have side projects that we're working on where you can add in some targeting to it to hit receptors of other different cell types, but not for vaccines. We think this is as good as it gets.
Good. That's a very nice definitive answer. Next question, how do you make the nanoparticle? Just mixing the compound with mRNA, and how does this platform protect the mRNA and enhance transfection and endosome escape?
Okay, there's quite a lot there. Essentially, the RALA peptide comes in a lyophilized form, and the messenger RNA is in an aqueous solution. And essentially, we actually used controlled rate microfluidic mixing, and we don't use any buffers, so it's purely molecular grade sterile water that we mix them in. Let me see what the next part of the question was. Okay, so how does it protect the messenger RNA? Well, that's quite easy, because the peptide intercalates with the messenger RNA, and essentially along one sequence of the messenger RNA, there are on average 695 pieces of the peptide that basically link in to that messenger RNA, and they form sort of hydrogen bonds with the gonado-head groups of the peptide, and this is on the phosphate backbone. And so because of that, it is really a complex intercalation. You also have got a cysteine residue at the end of the peptide that will enable the two peptides together to form the disulfide bond. So that really does give it an element of stability. The reason that it protects the messenger RNA is literally because the peptide appears to be like any other sort of amino acid sequence that is found in the bloodstream. We can't find any protein corona for it. In addition to that, we have found it to be stable in plasma and stable in serum. So it is quite interesting. How it enhances the endosomal escape, well, that is the pH responsiveness of the peptide. So at a pH of 7.4, the peptide basically is quite random coils. So the unfolded sequence of the peptide intercalates with the messenger RNA. Once inside the endosome has a drop in pH, and the RALA peptide becomes more alpha helical, so more structured. That fuses with the endosomal membrane, and it really allows full escape of the cargo.
Great, thank you. Was there antibody measured for E6 and E7 after 2 immunizations or in the therapeutic experiment?
Okay, so yeah. So it very much depends on the model that you use. So we obviously used a C57 BL6 mouse model to look at the T-cell response and to also look at the therapeutic response. In biopsy mice, we do get E6 and E7 IgG, and similarly in rats, we would get E6 and E7 IgG. So it is kind of really important to make sure that you pick the appropriate animal model for what you want to look for. So if we had have looked for E7 antibodies in C57 mice, we wouldn't have found them, and we may think, oh, it doesn't produce antibodies, but that's purely because that mouse model isn't designed for that.
Great. What pH buffer do you use for preparing the complex?
Basically water. So it's fairly straightforward. And essentially, once we make the complexes, as you know, we lyophilize it then with the sugar, and the amount of sugar that we add is important because that then enables, upon reconstitution with more water, to form an isotonic solution that we know won't cause any lysis of anything.
So that's Irish spring water, I take it.
I think it is Irish spring water, yeah, definitely. That's filtered,
Okay.
sterile.
Sterile, of course, yes.
pH 7.4, typical water.
Very good. This, as everyone watching knows, hopefully, is the fourth in our mRNA series of webinars sponsored by TriLink. And somebody asked, what's the relationship, if you don't mind me asking, between Phion and TriLink?
TriLink have been our preferred partner from day one to provide our messenger RNA. Now,
Yep.
we've worked with TriLink. Obviously, you know, this is our first product going into clinical trials, and we've been very impressed by the level and commitment of TriLink to help guide us in what is the best type of messenger RNA to get, and also the quality of the messenger RNA that's come through. We get very consistent responses. And now as we head into the clinic, you know, we have our GMP slots booked, etc. And they're our provider of our key cargo.
Great. Do you collaborate with other academic institutions?
Yes, we do indeed. We have a number of collaborations. Obviously, you know, I'm from an academic background and still within Queen's University Belfast, but Phion collaborates with Royal College of Surgeons Ireland, Dublin City University, Trinity College Dublin, Penn State. We also collaborate with Birmingham University. There's a whole range of them that we collaborate with, because we're keen for people to try the technology and see how it works in whatever it is they want to do.
So where this question may be leading is, are you open to additional academic collaboration?
Absolutely. We
Okay.
you know, we don't claim to know every application of this technology, so we're always keen to see if other academics might be interested in trying this for whatever needs they might have.
Helen, you're juggling two very, very big roles, professor at a leading academic institution in Northern Ireland and as CEO of the company. That can't be easy, I don't suppose.
No, but Phion is a consequence of, I suppose, 15 years worth of research. So it's a lifetime of work, I guess. And when I started my research group in 2006, I really wanted to make sure that, you know, if I did build a delivery system that worked, that we could do something with it. And it's a journey that I want to continue to learn on. I mean, you know, I don't want to essentially sit in an ivory tower and just produce papers, etc. I really want to continue to be challenged. And believe you me, Kevin, in this role, I am definitely challenged.
Great. Well, let's get back to some of the technical and scientific questions. Have you done a side-by-side comparison for immunogenicity of RALA mRNA with LNP encapsulated mRNA for benchmarking and naked mRNA as a negative control?
Yeah, we've definitely done studies with naked messenger RNA. We haven't done studies with head-to-head with lipids. However, I'm sure everybody that is aware of mRNA vaccines will know that a lot of really impressive companies have published their preclinical data, which is totally invaluable for, you know, young SMEs to see what the benchmark is. And I'd just really like to thank those companies for doing that. And the reason why we haven't done it ourselves is because we are not experts in making lipid nanoparticles or LNPs. So you know, the lipid nanoparticles that we would make would not be of the standard that we would need to compare against. So what we do is we know the levels that we need to hit because other companies have shown what they can do. and we basically benchmark across that way.
Mm, mm. How do you produce the PTX-V4 mRNA? Do you produce one mRNA or 3 mRNAs and then mix them?
Yes, so PTX-V4 is very interesting. So we produce the 3 individual messenger RNAs because we want to check, functionality of them all, and we can mix and match them. So we actually mix the 3 messenger RNA together, and then through automated production, we make a nanoparticle that contains all 3 messenger RNA. What is really key on the back end of the production of these particles is essentially making sure that the nanoparticles that we test have the 3 messenger RNA in them, and to also make sure that those messenger RNA remain functional. Some people might ask, okay, well, how do you decide on, how much of each messenger RNA to put in? You've got to really base that on the mass of the messenger RNA and the copy number, and that will really determine. And also, if you've got one antigen that's dominant over another's, that may change your ratios. But you have the flexibility to put in 3, 4, we've even put 5 messenger RNA in a nanoparticle, and you could put in more if you wanted to, I guess. But we have the flexibility to kind of decide what our quantities are and maybe have a little bit less of one and a bit more of another.
Okay. What is the relative weight of the RALA peptide versus mRNA in your formulation?
Okay, yeah, so it very much depends on obviously the size of the messenger RNA, but we do know that on average in each of our nanoparticles, essentially we have got somewhere in the region of, let's say it's 1 messenger RNA, and again this very much depends on the molecular weight, but let's say we're looking at a messenger RNA that's around 750 nucleotides. We would get around 1,200, 1,250 copies of the messenger RNA per particle, and it's about 880,000 copies of the RALA per particle. Now, the ratio that we pick is something that we decide at the start. So the ratio that we may be using could be what we call an NP ratio, which is essentially the number of amines in the backbone of the RALA peptide that is required to neutralize the phosphates in the backbone of the messenger RNA. And once you determine that ratio, so say for example 1 microgram of messenger RNA might be neutralized by 1.4 micrograms of the RALA peptide, then that is your NP1 ratio. So if we were to use, for example, an NP10 ratio, that would be 14 micrograms of your RALA peptide to 1 microgram of your messenger RNA. We actually use a ratio in our vaccines of less than that, but that gives you an example of how it works.
Okay. As you can see on the screen, Helen, we've received literally dozens of additional questions. We're not going to have time to get to all of them, or even most of them. If there are 1 or 2 that you would like to pick out from that list that catch your eye, by all means do so. If not, I can take a couple. And I have another question, just a broader question, which I'll start with, which is, obviously we've all, pretty much everybody watching has benefited from the rollout, the rapid rollout of RNA vaccine technology for COVID over the last 3 years. I'm wondering what you and your company and your colleagues have learned from everything that we've seen in the clinical and commercial development of mRNA vaccines for COVID. What can you learn, and how can you benefit from what you've seen over the last 2 or 3 years?
Well, I think what we have seen very much is that, no one company or one entity, for example, can work in isolation, and I think that's a really nice thing. I think it's really forced the key scientists across the world to come together and collaborate, and I think that was probably missing pre-pandemic to a certain extent. I think the value of science has been wholly recognized throughout the world. I think that's a very important thing. But I think an openness and a willingness to find a solution to these key global problems as well is very important. So we have learned as a company that there's an awful lot that we cannot do ourselves, and that's fair enough. So what we do is we reach out to key stakeholders who have expertise in the areas we don't have, and we really rely on them. And we also adopt a very open, transparent company whereby we're very keen for people to come and talk to us. We're very keen to work with collaborators, scientists, academics, just really wanting to try to achieve the end goal, which is to make these messenger RNA therapeutics, the gold standard of the future. I think pre-pandemic as well, it was very difficult to raise awareness about the technology of messenger RNA. There was many scientists working on this for decades, but yet it didn't really translate into a therapeutic. It's been really nice to see that now the floodgates are opening. And, it's funny because the media would say this new technology. I think for those of us working in the field, we would be saying, well, actually, we've been working on this for many years. You're just now starting to listen. And so from that perspective, I think an openness and willingness to try new things has also been really important.
Great. Are there any questions from this list, Helen, that you would like to take?
There are a lot of questions.
There are an awful lot of questions, yeah. Pick a couple at random and before we close, and perhaps if it's okay, if you don't mind, and you're open to having folks either contact you directly if they don't get their question asked. Obviously, there's going to be contact information on the FION or Helen's academic website. I think the level of interest is such that I'm sure you wouldn't mind spending a bit of time answering these offline.
Absolutely not, definitely. Will we have one?
Okay. Yeah, go ahead.
Take a couple if you want me to.
Go ahead, yeah.
Yeah. Okay, so we have one from David Holler who asked the question, was there any extraneous biodistribution expression in the tissues of the PTXV4 hamsters? That's a really great question, David. We're actually expecting the full histology and all of this coming back mid-November. And actually, as part of our toxicology study for PTXV1, we will be looking at messenger RNA distribution in a range of different tissues. But with respect to biodistribution when we inject IV, our typical sites are actually in the order of lung, liver, and then spleen. And there is also a perception, for example, that, when you deliver something intravenously that it's got a high cationic charge, it's definitely cytotoxic. What we've discovered is that actually zeta potential is really not the good measure of whether something is cytotoxic or not. It's actually the charge density. And the charge density, as you can see in the sequence of the RALA peptide, is spaced out quite nicely. And because of that charge density, we get a charge density rating of less than 1 whenever it's complexed with our with a cargo. On its own, it's sitting at around 1, and anything really less than around a charge density of 2 really isn't toxic. Let me see. We have somebody from the University of Pennsylvania, CETA, I believe. They asked the question, what is the ratio of peptide to messenger RNA? Well, in these particular studies, it is an MP ratio. I can't give you exactly it away, but it's typically an MP ratio of somewhere in the range between 6 and 10. So I can narrow it down that bit. We actually also have a question from a chemist that I work with in QUB, Joe Val. He asked, is there any issue with different oxygen levels in the water, keeping the disulfide levels consistent? And in true fashion, I actually don't know the answer to that question because we haven't actually measured it, but it's a really good point, yeah. And then we have, here's a good question as well. We have a question from Carla Belmonte from UNC, and she asked the question that the particle applications go beyond the vaccine generation, but also as a delivery system of other therapeutic messenger RNAs. In that sense, have you checked your nanoparticles in the brain? Do they cross the blood-brain barrier? We have actually looked at this, Carla, and we get a very small amount that crosses the blood-brain barrier, and it seems to cross very quickly within an hour. And so I think that you would actually need to have some sort of other targeting motive on this to get across the blood-brain barrier. So it's not something that readily happens. It's just a very small amount, probably in the region of 2-fold over some of the other sites that we see.
Helen, I think we'll leave it there for questions. I know we just we're never going to get through the bulk of these, but I very much appreciate you offering to handle and briefly address any pressing questions that you have for Helen. If you contact her offline, she I think really appreciates the interest that you've all shown audience to her presentation, and she'll take those offline. So we really appreciate all of our speakers for doing that. So Helen, thank you so much. That's the close of today's webinar. It'll be available on demand shortly for repeat viewing and sharing with your colleagues, along with the other 3 episodes in this excellent series on mRNA, sponsored by TriLink, which are already available on demand. So do look for those at the GEN website. Many thanks indeed to Helen McCarthy for an absolutely superb and very popular presentation by the looks of things. We thank also TriLink for supporting this webinar series, and Jeff Bagaliskis, the GEN Multimedia Group, and Workcast for all of their great work in hosting the webinar. But most of all, we thank you, our audience, for tuning in today. So for everyone at GEN and the CRISPR Journal, I'm Kevin Davis. Thanks for watching, and goodbye for now.